Okay, sorry. Well, thank you very much for the invitation to speak. And one focus of my group is statistical methods for analysis of expression data, especially with implications for cancer studies. And so luckily, I'm lucky that on Monday, I believe it was, Angela actually spoke about alternative splicing and implications in cancer. And that's a nice biology introduction for me, so I'm not going to spend a lot of time on that. But just to make sure, in case you weren't there, what I'm going to be speaking about is alternative splicing. And of course, what I mean by that is the process where the pre-mRNA can be has introns that are removed from it, and this can be done in multiple ways that can result in ultimately different mRNAs that can therefore then be processed into different proteins, for example, or it can be more subtle. Different mRNAs can have different likelihoods of being degraded and have other kinds of effects that don't always translate directly into proteins. And this process of alternative splicing has shown up in tantalizing ways in cancer literature, and Angela spoke about the efforts and her efforts and others to try to parse out what are these implications. But just to give an example, in AML, you have the UTAF1, which is a splicing factor, which is significantly mutated. In lung adenocarcinoma, it also shows up. Mutations in SF3B1 are very common, even more common than they are than these. The prevalence is higher in uveal melanoma and also in CCL. And so these things are showing up, and so there's an interesting question as to what implications these splicing has. So what I'm going to talk about is more our efforts in terms of the methodology side, in terms of statistical analysis of expression data and what kind of things we can do in these types of studies in terms of methodology development. So the first one links directly to the type of analysis that Angela spoke about on Monday, and in particular looking at exons and which exons are spliced in and out. So just to remind you of that type of analysis, so if we have mRNA-seq data, for example, from samples, there are different types of quantities we can be interested in. Obviously, the most basic one would be the overall gene expression analysis, which we've been measuring for ages since microarray data is earlier. But with sequencing data, of course, we can measure other types of quantities. We are methods to try to estimate the individual isoform level expressions. We can also look at junctions, splicing junctions that indicate what parts are being spliced in and out. And what we're going to focus on is the similar type of analysis that Angela mentioned, where you're really focused on exon by exon analysis as to what is being spliced in and out with respect to a particular exon. And this is particularly useful in cancer, where you have a lot of novel, or you suspect that you might have novel junctions or novel types of isoforms or products that are being created because of the disruption of the cell. So just to be more specific, if we have a little cartoon example, we have a set of exons. There are a certain number of junctions that will indicate that the exon was included, and there'll be some junctions that indicate they were skipped, and so we can have some sort of basic, if you like, count matrix where for each exon you have the number that include and the number that exclude. And what this would, of course, generally be summarized by some notion of the percent spliced in, where you just take this as a percentage. And we want to ask the question, are they different between groups? This could be tumor versus normal, but it could also be one subtype with the mutations versus other samples, and so forth. And so we're interested in the question of statistical methods for how to best look at this question. So, of course, gene expression has a huge amount of types of methods, and the standard sort of gene analysis framework that we have going back from microarray data is to look at each individual gene separately, do some kind of t-test, and then find the genes that have significant marginal relationships, which is very early on. There were statistical challenges that were addressed with the small number of sample size that could often occur. You have large number of genes and you're making a lot of estimates. So you have problems of false discovery corrections, and also instability in your estimates that mature methods try to account for by using various types of regularization, and in particular, empirical Bayes type strategies. And just continuing with the gene example, of course, when there was microarray data, you had roughly normal data. Sequencing technology gives counts. And so there's a huge industry of adapting these, what we already know works well in microarray data, to count data. So first, what's the distribution? You could say it's Poisson, but that's not a very adequate model for the data. You usually want to add additional dispersion, what's called dispersion, which is like variance, the ability to say that the samples are more variable than you would see with a Poisson, and then questions of shrinkage or a regularization of your estimates. And there's just a slew of methods that do this in GeneArray. EdgeR, VOOM, DE-seq are big ones, but there's probably 20 or 30 different proposed methods that basically do the same strategy in different ways. So just going back to the exon usage, so what we're looking for is sort of to be able to have a similarly mature type of analysis for this particular setting. And what is different? Well, we have two sets of counts, if you like. We have the counts that include and the counts that exclude. Or we could rewrite this as the number that include versus the total, and what we're looking at is the fraction. So clearly, just from a starting point, Poisson doesn't even make sense here as a starting point. It makes much more sense to think of this as a binomial. These are proportions we're trying to estimate. But we would like to keep the same kind of strategy. And so there's not so many methods that exist that are applicable, and most of them are not created for exon usage either, but for different settings. And so we develop a framework for this over-dispersed binomial data by picking a particular way to add this dispersion parameter and then providing a shrinkage estimate. And as I'll discuss, it has some nice generalizability to other types of settings beyond just the exon usage, which we think is also nice because there's other alternative splicing questions that might come up and imply different settings. And so it gives us a way to expand into other settings quickly without having to reinvent the wheel every time. So what do I mean by an empirical Bayes strategy, just to make that clear? Well, we could start, if we have some data, again, on our setting, if we were using, for example, Bayesian estimates, we would say, well, our data has some distribution if we know the parameter, and we're going to assume the Bayesian side of it would assume that parameter actually itself comes from a distribution. It's random. And we would estimate that parameter as the posterior mean of the data. And Bayesian estimates have some nice properties, that they have this sort of regularization property that they stabilize their estimates, if you like, and they sort of shrink it towards your prior. And moreover, this shrinkage is implicitly controlled by the method. There's no tuning parameter. It's controlled by your sample size. So with larger sample size, you shrink less towards your prior. But of course, you have to pick your prior, which is an arbitrary process. But so empirical Bayes strategy is trying to merge this Bayesian method with using the data for the prior estimation. So specifically, if I'm thinking I'm doing this same modeling on thousands of genes, I'm not going to set up a different Bayesian model for every gene. I'm going to use the same Bayesian model, and I'm going to just repeat thousands of times. And so what that means is that I'm assuming I have the same prior for every parameter. So it may be it's different, for every gene, but it's coming from some overall distribution. They can't be that different from each other. There's some restriction. They share something about them. And so given that I'm doing this thousands of times, I ought to be able to look at my data and say something about that, not just have to pick it arbitrarily. And that's the idea of empirical Bayes, is that I've done this so many times that if I can look at the marginal distribution, integrate out the parameter, and look at the marginal, then I have a lot of information about my prior, which I can think about as the parameter alpha. And once I know that prior, then I can then calculate with this data prior what my estimate would be. And so you get the shrinkage towards the data now, the overall bulk of the data across exons and genes. And so this is like a likelihood method for shrinkage, and again, doesn't require any tuning parameters. In general, this is actually difficult to do what I just described exactly the way I described it. In particular, if you don't have nice distributions that play nicely with each other, these processes can be difficult. So in particular, we have multiple parameters going around, not just one, and so we want to have different treatment of, say, the mean versus the variability. And then also, depending on what distributions I'm picking, it's not always easy to work with priors and create empirical-based strategies. And in particular, in negative binomial, which is what's used in the gene world, this is a big hurdle, and so there's a lot of other sort of pseudo-strategies that try to get at doing this basic idea. So what we do is we pick a particular choice of a dispersion model, so a particular way to take an initial model like a Poisson or a binomial, add dispersion to it so that you have this variability that you need to capture population dynamics. And this is a method proposed for Ephron, and it's general, which is nice compared to some of the other strategies that are usually used. It can be used for a normal, a binomial, a Poisson, a multinomial, anything in the exponential family. And so what we show is that, in fact, if you look at the maximum likelihood estimate, it's approximately gamma distributed. And moreover, so by doing this, we can develop an empirical-based strategy so that we can then use conjugate priors and go forward and actually get an analytic solution for how we would shrink our data or regularize our estimates with respect to the rest of the data. And so this is, we can use this for, again, any distribution, but again, we focus on the binomial because we're interested in the exon usage case, where we're looking at proportions. And just to give some idea, there are a few other methods, not so many. I can put them all on one plot as opposed to gene estimates. And so looking at simulated data, it's striking that most of the other methods do not control the false discovery rate. So in particular, you can have a reported false discovery rate of 10%, and actually you're getting somewhere around the order of 40% false discovery rate. So sometimes it can be really quite large, what the difference between them. And this is not actually simulated under our model. This is simulated under their models. And we also have relatively high power that, in terms of the rank of the discoveries we find, tend to be we have to go pretty far down our list relatively before we start finding false ranks compared to other techniques. And we can also, so then we also look on real data. Sorry? Oh, I'm sorry. That's an important part. I'm sorry, I forgot about that. My method is the black line here, the web-seq. So the one that follows, so the black line, the one that gives you a low false discovery rate and gives you a relatively low ranking of when you find your first false discovery. Yes. And the other one that has sort of some high power just has very, very high rates of false discovery. So, so they can find some things, but you don't have any confidence in it. Say again?
You rank by p-value. Where do you get it from?
Oh, so I didn't talk about that, but then I go back in and I use these dispersion estimates, and now I test between the groups, between the means, using likelihood ratio statistics based on the model.
Why not just base factor?
It's sort of the same thing. It's a likelihood ratio statistics. It's not going to be so different, but yeah, that's what we use as a standard test. We looked at different tests, and they do have impact on how well they perform. But yeah. Sorry. And so again, looking at a more realistic setting where we actually create a null setting, so there isn't any truth in there, by randomly just permuting real data. And we find similar behavior in terms of the fact that most methods have very high false discovery rates. Although, I'm sorry, this is false positive rates, because we don't have false discovery rates in this setting. And then if we look, here we're actually comparing different, we were trying to make sure that there was something to find. So we're actually comparing two different tumors from different tissue types. So we're largely not picking up a tumor signal, but picking up a tissue sample, which we know will be large. And we find that, in general, most of the things we find are supported by other methods, but most of the other methods find a large, large number of things that are just not supported by any other method. And here I only show 4, but we actually compared to like 8 different methods. And even when you compare across all the 8 methods, most of them are just finding things novel to their method, even though they're assuming the same distribution and the same things, unlike us. So it's so it gives an implication that the things we're finding seem to be reliable. So the other thing I didn't mention is that you can also use the same kind of analysis looking at exons by using isoform counts, and so that's the same idea. And as I mentioned, it extends to other distributions easily. So for example, if you started with a Poisson instead of binomial, you could do gene expression. And we don't really want to get into the gene expression fight because there's I've shown only a handful of the many things out there. There's a lot of methods for gene expression, but just as a point in principle that it expands well, we again control the false discovery rate pretty well, even though this isn't our distribution, and we have pretty high rankings in terms of the things we find. More interesting is that you can then expand into other settings. For example, you could look at the multinomial and try to be comparing isoform proportions rather than just per exon and things like this. And it gives a straightforward, nothing changes, everything stays the same regardless of the distribution. Okay. And so then the other area that we've been looking at is something that's particularly relevant for tumor settings is actually novel subtype. Rather than starting with groups that you want to compare, trying to find clustering. And how does alternative splicing impact or influence that process? So in general, when we're clustering people, we have isoform estimates from mRNA-seq data, but we're still largely, I think, using gene expressions, for example, in many of the TCGA papers. And that's probably not generally a very big problem, we would think. In fact, in many genes, if you look, for example, across individuals and at the gene expression, if you then broke it into the isoforms in that gene, you would have many genes that might look like this. There's not really a large pattern difference of how they're performing. However, we do know that there are some genes, that these genes exist that do have isoform differences. So for example, we can look at a different gene and we look at their isoforms and we can see that in a subset of samples, there is a real switching behavior as to which isoforms they're using relative to the other samples. And this doesn't show up from a clustering, if you were trying to find them in the gene, you might find it, but it's not a very strong signal here compared to what is a very striking signal here over in the isoform estimation. If we look more broadly across many isoforms, if we look, for example, in the TCGA AML data, if we look at the subset of variable isoforms, and then we take those same genes that they came from and just look at their gene expression estimates, some of them don't change, right? Like this blue group doesn't look so very different here in the gene expression versus the isoform, but there are some groups here that have strikingly different patterns when you go from one to the other. We're missing some signal when we make that transformation into the gene world. And similarly, if we look at the UVM data, we can find similar types of patterns where, again, a lot of it stays the same, but we have, again, it just happens to be colored the same. It's not the same group by any means. But we have a diminution in the signal here. And in fact, we have this set of samples that seems to look very different here in a way that they didn't in the isoform. And in fact, when you cluster, the gene clustering picks up these samples in a way that the isoform clustering does not. But clearly, when we're talking about clustering, it's a numbers game because clustering is driven by these large groups, and we have thousands of genes. It's driven by these large groups of co-expressing genes in bulk. And so what effect it's going to have is not clear because you can be missing the signal on a lot of genes and still manage to cluster perfectly well because there's so many other genes, perhaps with the same signal, right? So it's a kind of question mark as to how important it is. So for example, So we sort of look first just to see what kind of differences do you see. The AML paper, oops, sorry, wrong button. The AML paper, when they find their clusters, they look at spliceosome, for example, and they don't find any particular noticeable patterns. When we look at the same data, we look at specific genes, and we do seem to see more patterns in the sense that the UTAVF1, if we accumulate all different kinds of mutations and copy number changes, do seem to cluster well, as do these. But we'd also note that we find those same patterns pretty much in the gene. And they could very well be related to just the fact that these are also samples that have complex rearrangements, and there could be other things that are driving it that we're picking up as well. And I would also note that this is different than the paper. We're not recapitulating the results of the paper. So that's also a caveat, although they make biological sense on other respects. And similarly, if we look at the UVM data, I'm sorry, I meant to say one more thing, is that, so they're generally pretty much the same, although it is interesting that you have one particular fusion gene that is clustering into 2 different groups depending on how you cluster it. So it is making an impact. And similarly, if you look at the UVM data where they have a fairly highly prevalent mutation in a splicing factor, you seem like you probably are getting a little bit more clustering of that splicing factor into groups, not the same groups, but 2 different groups versus the middle compared to the gene. So it at least suggests the question that there might be some kind of overriding splicing patterns that could be detected by looking at the isoform. So then the last thing I would talk about is some ideas we have been working on trying to really look at clustering from this point of view. What is there shared in terms of splicing in particular? And so what do we mean by this? Well, the way we've been thinking about this is rather than clustering isoforms and just throwing them into a clustering technique on their own, can we put them in as a group, so to speak, and cluster on them based on their group behavior. In other words, how they perform relative to the other isoforms. So rather than just taking these 2 isoforms, clustering them separately, do we get some advantage by looking at them as a group and their relative performance? In particular, we could look, for example, there's the proportion, and we could convert these into proportions, and we would have one number that's a proportion, and we could use that instead. Now, obviously, we actually tend to have more than just 2 isoforms in many of these genes, and so we can look at other isoform expressions. But we can still look at the relative proportion and try to use this as sort of a proxy as to how it's performing relative to the other samples and see if we can get different types of signals when we cluster on them. So what do I mean specifically I'm going to do? Well, for each, rather than having, say, genes by sample, which is the standard data input, what I'm going to have is genes by samples and then another dimension on my array. Only it's going to be a ragged array because I'm not going to have the same dimension for different rows because isoforms will have different numbers of, genes will have different numbers of isoforms, so it won't be the same set. That's not a very attractive input for most clustering algorithms. So the strategy we take to try to perform this clustering is just to think about each gene separately. In particular, for each, what does a clustering algorithm really usually wind up being is some kind of distance between samples. And so instead of trying to tackle this thing directly, instead what we take is we look at each gene, and creating a distance matrix for each gene is not difficult. There's a lot of ways of comparing distances for vectors that are on a simplex, for example. And you can entropy distance, Hellinger, there's all these different things. And we can get a different distance for each gene and then try to combine them. So standard clustering algorithms basically do this anyways, and they just take the average. You could imagine using a weighted average, and we have tried that and not had a whole lot of, and you could imagine trying to find the best weights from the data, but we haven't had a lot of luck. We get very variable and unstable results, so we haven't really pursued that any further. So the question could be is, what more do you get out of grouping them together versus treating them separately? Because implicitly, all that information should be in the isoforms, so why not just cluster the isoforms? Shouldn't you be able to get it back? So the first thing, that was the first thing we were asking when we look at simulated data, at least, which I don't, you don't want to push too far, but at least it gives you an idea that there is some information or additional sensitivity you get by grouping them. So in particular, if we look at the similarity to the truth, we have the isoform method here, and we have methods that work by grouping the isoforms into genes, and we can see that we get, improved performance on the simulated data. So at least in principle, you can find things that you might not be able to find as well if you were looking in the clustering data. So we look, for example, in the work on that Angela presented in finding differences. And if we just isolate ourselves to the set of genes that they find have significant differences, we are able to recapitulate the cluster. It's not the first break, but it's pretty close to there. Another thing that we can do is if we look at the AML data, we can just try to apply this to all the AML data. So our first round of doing this was a success in a certain sense. We very successfully identified a batch effect. We identified it really precisely in the sense that if you, these are different clustering techniques. This is the gene, the isoform, and the proportion. And this is the plate in which the AML were on. And so we almost perfectly recapitulated the plate effect using our proportions. The isoform finds it to some degree, but not so nicely, and the gene eventually starts to find it, but it's not, so to speak, a priority. And so does this have anything to do with alternative splicing? Well, maybe. In fact, if we look at the 5' to 3' coverage, that suspect plate looks very different from the others, which could really affect your isoform estimation procedures if you have a different kind of coverage over them. This was not reported in the AML, but we have some question marks because I think they did some kind of batch correction. So that's a gold standard of a sort. It's not very interesting, but at least it seems to work on large scales in that setting. So then we were interested in doing, after we normalized the AML data, and except for this one slide, everything else I demonstrated was on normalized, batch corrected data. So if we look at the batch corrected data, what kind of information can we find? And we can find that we do get some different clusterings from the proportions. In particular, this is the cluster identification of the samples, and these are the different isoforms across the rows. And we can see that they are highlighting two different things. In particular, if I look at the proportions, they seem to be matching much more these sets of isoforms compared to, for example, some of these other isoforms that perhaps match more with the isoform clustering. Again, it's a numbers game. It's kind of where is the focus going to be in the clustering. Well, so what kind of thing is happening here? When we look at some of the genes that we see there, there's a strong switching pattern where there'll be one isoform that's in, say, the green group, another one that's in the blue group, and we can see that there's a strong switching pattern that is creating different clusters. And it seems to be shared in this behavior. In particular, what does that mean more generally? Well, how does this look like when you look across the genome, across the transcriptome? In particular, if we look at this blue cluster, which doesn't match our clustering, a lot of their genes sort of match that description I showed before. A lot of their isoforms match the description I showed before, that there's not a lot of difference across the gene. In fact, if I look at the isoforms that are in this blue cluster, and I see how many of the other isoforms that are in its gene are also in the blue cluster, it's about 70%. So most of these isoforms, all of the isoforms from that gene are also showing up in the same cluster. So it makes sense that they all have the same expression pattern, and that's not something that's interesting to the proportion. But if instead I look at the green and this cyan color, we see this switching pattern I showed before, and in fact they are co-occurring at sort of significant rates. They're much more co-crossing in the sense that the gene will have one isoform in one group and the other isoform in another group. And that's what we're picking up from the proportion clustering. And so, in general, it seems like there may be widespread splicing patterns. I wouldn't go so far to say they are, but there are, at least looking at isoforms and looking at the mutations, it does seem to be that there is some possible signal there in a more widespread nature. And that we think by clustering on the isoforms as a group in a gene, for example, by taking their proportions or in other ways, can sort of give an orthogonal look in terms of the priority you give in your clustering between the isoform and the expression patterns. And just to acknowledge my students, Sean Reddy, who worked on the first part about differential exon analysis, and Marla, who's been working on the clustering and the data from the Cancer Genome Atlas. Thank you.
Thank you. Do you have a question? No.
I'm sorry, I was just...
Okay. Yes?
Do you see differences in survival in any of the new clusters that you see with the isoform model?
We haven't gone down that. I would be surprised, frankly, if there was. But I mean, I feel like it's a piece, but I don't know. It's something we've been meaning to do, and I think we looked at it briefly, but I couldn't say what the results are, so I'll just say we haven't done it. I suspect we've looked at it, and it wasn't particularly interesting, but yeah.
I wonder if you've looked at how the different methods for alternative splicing perform as coverage is varying. Do they have? Oh.
Sorry, I thought you were going to ask me a different question, a more downstream question. Well, I was going to say that we initially started by making our own isoform estimates, and then we switched over to TCGA estimates, and we see a similar behavior, and they're two different algorithms and run from. Oh, I'm sorry. What are you talking about?
In the first part of the talk, where you were just trying to predict alternatives.
Oh, I'm sorry. Oh, okay. I thought you were talking about the batch effect with the coverage. So you're saying how does the different...
Do the different methods for determining alternative splicing, do they perform differently in different coverage regimes? So are there perhaps methods that are much better at low coverage as opposed to high coverage? Because there's different ways you could use the signal.
Oh, okay. So you're talking about like basically the overall count level, the depth. Yeah. Oh, I'm sorry. I thought you meant how it was distributed across the gene. That's why I was going to the batch. Well, I would think that probably as you get high coverage, some of these questions become a lot less important. Right? As soon as you're so one thing I would say is that, for example, if you're looking at the splicing junctions, you have much lower counts than you would looking at gene or isoform or anything else. So you're in a very, very low count level compared to anything else.
Also the exon counts as well, right? So if it's the exon short or long, then...
Yeah, so that depends which way you do. You can do it both ways. You could include the exon counts or not. And if you include the exon counts, then you get a much higher coverage. But some people aren't super comfortable with doing that and prefer to look at just the junctions. I think we've done it both ways. And I think actually what I'm showing here includes the exon counts, actually. But having said that, so I think that there's a longer way to go for exons to get to high depth, but at some point when you get to high depth, a lot of the nuances about we need to use a count distribution and be careful about it and so forth makes a lot less difference. In particular, if we're just talking about gene expression, if you have very, very high counts, taking the log and doing standard microarray stuff will be probably okay. It's with the low count regimes or when you have low count in one side or another that it matters. So, but for the exon, I think you're so far away from the gene level of counts, that I think that you would have to have a significant bump up to not have that influence. In terms of the different count methods, I don't think that we've played that, but I don't think we saw any relationship between the overall, like which ones we were finding false or positive related to the overall count. So that's sort of the closest. I don't think we explicitly like downsampled everything or upsampled everything, but in terms of looking at the exons we had, which had a pretty large range of coverage, I don't think we saw any bias there with respect to that.
Did you look at all at mutations that were correlated with the isoform clusters and different isoform patterns?
So, I mean, the main mutations we were looking at were ones in the spliceosome, which were the ones that I was showing in the pictures. But, if you look, trying to see if I have a picture of any of them, but, so we have other features. The mutations, for example, when we were looking at AML, we tended to focus on the ones that from the AML paper and other things. I mean, it's very well characterized. And so we tended to focus on those, which in AML includes a lot of fusions, for example, and some deletions and amplifications. I guess this is a mutation here. So that was our focus. But in that sense, yes, we did look at that. And interestingly, the particular splice mutations, and there's only 7 of them. It looks like there's a lot, but most of these are copy number amplifications or deletions. But there's only, I think, 6 or 7 samples that had the mutation. And so while when we looked at the specific set of genes that a priori should show the difference, then we get a clustering of those particular splicing. But when we look overall, looking at the proportions is not improving the splicing of those particular mutations.
So if you just looked across all possible mutations, you didn't find anything that stood out as being...
I have to be honest, I'm not sure that we looked over all possible mutations. I think we looked at the mutations that were found significant and from the paper output.
Is the heterogeneity of the tumors an issue? If you have a single tumor that might be completely different isoforms and so on, is that something that can be addressed? Is it addressed?
I would think it would certainly be it certainly could be the case. I don't personally know a lot of knowledge about the diversity of isoforms within a tumor. They may exist. I've not seen a lot of work of that. I would think it would make a difference. Yes, it would be but it's hard to know exactly in what way. But I would certainly think so. Yeah.
could look for mutations in the RNA-seq data that correspond to mutations in your phylogeny tree.
I'm sorry, look for mutations in the, I didn't hear.
RNA-seq reads that correspond to mutations in your phylogeny tree.
Well, that's true, yeah, and you could look and see and try to parse them back with the isoform. Yeah, if you're lucky. if you're lucky, you might get some information, but I think it would be pretty sparse as to how many genes that would give you very much information about. But.
Okay, we continue at 11 a.m. and that's 10 more speakers.